Okay, cell metabolism is we're looking at all of the cell reactions, and it's the breakdown and the building up of different molecules and using energy and releasing energy, and all those reactions is what makes up the overall cell metabolism. And it's the cell is the site of an enzyme-catalyzed metabolic reaction. So all these reactions occur within the cell. And what does enzyme catalyzed mean?
It means it's the activation energy.
Lowers the activation energy, so the reaction occurs faster. Yes. So there's two types, anabolic and catabolic. An anabolic reaction is larger molecules are constructed from smaller ones, and the process requires energy. So they're building up big molecules from small ones, and it requires energy to build it up. It requires more energy. Well, it requires energy. The other one releases energy. Catabolic, large molecules are broken down, and when you break the bonds, you're releasing the energy to be used somewhere else. And the reactions of metabolism are often reversible. So you can take a polysaccharide and break it down into the monosaccharides, but then you can take those monosaccharides and build them back up into polysaccharides.
That's anabolic?
That's showing reversible. So if you take a polysaccharide and break it down into a monosaccharide, then what would be anabolic or catabolic reaction? Polysaccharide to monosaccharide. Catabolic. They both can be reversed? Yeah. It's because you're taking a large molecule and breaking it into small individual molecules. If you took the monosaccharides and combined them into a polysaccharide, that would be an anabolic reaction. Yes.
Just take anabolic small pieces, that big one. Catabolic, big one, the small one.
The way I remember, I don't know if it might be a little odd, but cannibalism, you break down your own catabolic. That's how I remember it. No, you break down, they tear apart yourself or whatever. I don't know. That's how I remember it. It works for me. How do you want to remember it? Oh, we're not talking about hair right now. So anabolism, anabolic reactions. It provides the substances needed for growth and repair. So when cell and tissues want to grow and repair, they use anabolic reactions to build up the tissue or whatever they're building up. These reactions occur by dehydration synthesis, which means that a molecule of water has been removed to join the two smaller molecules. So maybe a hydrogen from one molecule and the OH group from the other molecule would break off, and then they would bind at that point. They would bond at that point where the hydrogen and OH group was. And so the OH and the hydrogen would then form a water group. And so the water is taken out of that whole thing. Does that kind of make sense? I think there's a picture. Hopefully there's a picture. Or I'll add one. We'll draw one. But you're basically taking a water molecule out so that a bond can form between the two molecules, the two smaller molecules. Polysaccharides, lipids, and proteins are constructed via dehydration synthesis. It's an anabolic process. To form fats, glycerol, and fatty acids bond, and there would be a water that would be taken out there, and between two amino acids, a peptide bond is formed. Two bound amino acids called a dipeptide, while many joined follow polypeptide. So we'll talk about peptide bonds when we get to like translation, transcription, protein synthesis. We form peptide bonds. Okay, so here is two monosaccharides. So you see how there's a hydrogen and an OH group right here and a hydrogen and OH group here. So the OH group here comes off. This OH group comes off, and the hydrogen from this OH group comes off. And so now this oxygen is bound to this carbon.
But
And so this, let me use a pen here. So this OH, actually, let's do something else. Let's use the highlighter. You can maybe see it better. This OH.
Can you use a different color? I can't see it.
I don't know.
Oh, thanks.
That OH and then this hydrogen come off, and this O, because they're bonding together. This is how they make a bond. So these two molecules are stable on their own, but now this water leaves, and the oxygen then is bound to this monosaccharide, creating a disaccharide. So this is, they formed water. Right there. Okay. So that's why it's dehydration, because you're taking out water from these two monosaccharides, and it's an anabolic reaction. Come on. Oh, no, it's going too far. Here's the same thing where I think these are they on your picture? Are they highlighted blue? Yeah. Was the last one highlighted blue on your pictures? The O and, yeah, okay. You just can't see it up here very well. So you can see, oops, this hydrogen, OH, these are all going to break off, making this oxygen right here bind to this carbon. This makes our fatty acid, our fat molecule, or our triglyceride, and then we form 3 water molecules from these elements. Still dehydration synthesis, or dehydration reaction. It could probably be called dehydration synthesis also, in case you see that terminology. Dehydration synthesis. Because remember from chemistry, what's a like A plus B making AB. So it's a synthesis reaction, but it's a dehydration synthesis because you're losing water. Okay. Catabolism breaks apart larger molecules into building blocks. So it'd be like taking that disaccharide, breaking off into the 2 monosaccharides. These reactions occurred by hydrolysis. Hydrolysis is where a molecule of water is inserted into polymer and split into 2 smaller molecules. So that water that we produced in our other reactions, do we need to move you two? Where the water molecules that was produced, now you're going to break that water molecule. Breaking the water molecule is called hydrolysis, and the H and the OH gets inserted back into that disaccharide to make them back into monosaccharides. So it's the reverse reaction of what we just saw. So here, this is a peptide bond it's showing, where if we go in this direction, this would be our anabolic. That's what this double arrow means. This is our anabolic dehydration reaction, because we're producing the water. But if it were to go in the opposite direction, this is our catabolic.
You can just put the water back into it.
Hydrolysis reaction. Because when we go this way, we have water. It gets broken up. We break up the water in order to make our 2 amino acids separate. Can I switch it? No, not quite.
So what are examples of anabolic
Like this is an example. What do you mean? I'm confused.
I don't know, a product.
Protein. When you make a protein, that's an anabolic reaction. But when the protein breaks down, like when you digest something in your gut, that's breaking it into amino acids, that would be the catabolic reaction. Can I switch it now?
Yeah.
Enzymes are what control these mechanisms, or these metabolic reactions, and control the rates. Enzymes are proteins that we said lower the activation energy of reactions, so it begins the process to proceed more rapidly, and they're called catalysts. We saw that in chapter 2. Enzyme work in small quantities and are recycled, so you don't ever run out of the enzyme. You're not using up the enzyme through the reactions. It's always, it takes that, breaks it down, spits it out, takes another one, spits it out, okay? It doesn't get used up. Each enzyme is specific, acting on only one kind of substrate. The substrate would be whatever it's breaking down, or whatever that's attaching to it or coming into contact with it so that it can catalyze that reaction. Active sites are on the enzymes that combine with the substrate and the reaction occurs, and those active sites are specific for one substrate. And then the speed of the enzymatic reaction depends on the number of enzymes and the substrate molecules available. So the more enzymes you have, the faster the rate can go, or the more substrate, you can have faster reactions. But there's a certain point. What?
It's not only
It's like the big space. I tried to reform. I missed that one. There are big spaces all over. So remember this graph that we had where we had the rate versus time? And it has, the more and more you increase the substrate, it finally just plateaus. Remember that graph from chapter 2? Where the rate, when you have increasing amounts of substrate, the rate will increase and increase and increase until we've saturated that enzyme and it can't work any faster, and so the substrates are still just kind of waiting in line.
But it could still go up, can't it, after a time?
No, the rate stays the same, and then once it gets done with all the substrate, then it would drop back down. It would drop off. It can only go so fast. So the rate would taper off, plateau. Factors that alter enzymes, so the proteins, just like the proteins that we studied in our lab and in chapter 2, they can be denatured. They denatured by heat, pH levels, chemicals, electricity, radiation, and other causes. There's some enzymes that are cofactors because they are like floating around. They're present in inactive states, and they need a certain cofactor that would attach to it to make it active. And the organic cofactors, they're called coenzymes because they work with the enzyme. So sometimes it's minerals, sometimes it's vitamins, those different molecules that are needed for the enzyme to become active. So here's two substrates. So this is the reaction that is trying to, well, would this be an anabolic or catabolic reaction, looking at, to go from A to C? Anabolic, yes, because we're taking two substrates and making one product. So we're taking these two substrate molecules. This enzyme has an active site right here that fits just these substrate molecules. They attach, it catalyzes this reaction, creates that bond, and get that product. Yes. It doesn't really look like that. It's just for... This is anabolic, yes. If we're going from left to right. Now, some enzymes break down. So if you went from right to left, what would that be? Catabolic. And we can't really tell whether it's a dehydration or hydrolysis synthesis or reaction because we don't know if it's water involved there. More than likely it is, but yeah. Okay, so this is one of the videos. Let's see what this is about enzyme action and the hydrolysis of sucrose. It is.
I have a question real quick.
Yeah.
That's
On the
cute.
picture in the quarter one quiz, is L pointing to the stomach, right? Or is that
I always wondered that too. I
it
thought
the
it was the
stomach?
liver.
I don't know
first,
what-
But I'm pretty sure it's the stomach.
It's like right
It's the stomach.
Is it the liver is like a big, huge
Yeah. It's
thing,
not
and then the stomach goes right under the liver.
It's the stomach.
Okay, let's see. Oh, I was gonna check my volume too. All this is going because get to work. I'll restart it if we can't hear it right away.
Sucrase resides on the surface of the microvilli on the intestine. As table sugar into its component monosaccharides, glucose or blood sugar.
Okay.
An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars.
it's anabolic.
Once this is accomplished, the enzyme's configuration is changed back to the original shape.
Catabolic.
The two monosaccharides float away, and the site
you
becomes
take
available
two
for
and make
another
one.
sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
Do you like the videos?
Yeah. How does it wear out? How does it wear out? Or
I'm not sure if it over time it only is good for, I don't know for sure. How do they come to become
Is
denatured?
that because of temperature?
If temperature, heat, pH, chemicals, those tests that you guys did on the proteins. Yeah, but it's not a denaturing chemical. And then there's another one about how enzymes work. Let's see what this one says. They take a little bit to load.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
And that would be the catabolic if it formed two products from a single substrate.
So catabolic, two things into one.
No.
No, and
No.
just think of it this way: cats tear things apart
Yes.
Do it. Sew it. Anabolic.
Yes.
Alright.
Okay. Energy is needed for the body's metabolic reactions. And the forms of energy include heat, light, sound, electrical energy, mechanical energy, and chemical energy. What would be an example of chemical energy that your body uses? Starts with an A. And it has a T.
ATP.
ATP.
Yes. What is? Is it?
Again.
Actually, let's just stop here. We'll stop here.